All right. Hello, everybody. Welcome to our first and only ever virtual class. So I will share my screen and we will get started. I hope you all have a happy Halloween. All right. So we'll start off. We're kicking off this section on nerves and nerve tissue. First, we'll kind of break down the organization of the nervous system itself into its different components. We'll look at the different structures that are involved with each component. And then not today, but later on, we'll look at how nervous tissue works and how action potentials are generated and how they're transmitted down the membranes. So first we'll start off with just like the structures and divisions of the nervous system itself. So you can think about the nervous system as primarily being divided into two separate systems, and each one of those systems has a series of subsystems that all, interconnect and communicate with each other. So we'll start off with the central nervous system. So this is the first one that forms when you're an embryo and you're going through the initial stages of development. The central nervous system develops out of the neuroectoderm, and we'll talk about more about that when we get into module 4. But this is primarily just the brain and the spinal cord, and that's about it. So it's like you can think about it like a long tube with kind of a bulb at the end, and the bulb is your brain. The other part of this is the peripheral nervous system. And so this one derives from the neural ectoderm, but it's after the neural tube folds, there's some cells that are kind of adjacent to it called neural crest cells. And they play a pretty significant role in laying down the patterning for nerves as they innervate different tissues as you go through different stages of development. And so the peripheral nervous system, it's basically, you can think about central nervous system is the trunk of the tree, and then everything else is peripheral to the central nervous system. So first we'll talk about like the different types of nervous tissue cell types. And there's a couple, and what's interesting is this is one of those things where location is going to dictate terminology. And so you can think about it kind of like this, like a taxi cab in the United States is called a taxi, whereas like in Great Britain, it's called a lorry. So depending on your location, it's going to have a different terminology, even though you're talking about essentially the same structure. So there's two categories of cell types. You can think about it like this. One is the primary nerve tissue itself. So that's the one that's generating action potentials. And then there's going to be the glial cells, which are the supportive tissue that they have a lot of roles, multiple roles, if you will. So they offer structure, support, nourishment, repair, that kind of thing. And so the first thing that we think about, so the two categories are, the first one is neurons. And so we can think about neurons as just being like the highway of traffic for communication within the body. So these are the ones that are transporting the signals from one location to the other. Neurons are going to have several different structures associated with them. These should all be familiar just from our section on nerves in module 1 in our first practical. But there's going to be a cell body. Sometimes that's called a soma. So soma means body. And so they have a cell body, and then they have processes. And so there's essentially two processes that extend from the soma of a nerve cell. And the first process is the dendrites, and then the second process is the axon. And as we'll see when we step into the different types of neurons, each one will come with its own set of processes that come off of the soma itself. And so, like I said, we have axons. So you can think about the axon as kind of being like the freeway. So the soma is where the actual processing of information takes place. But then axons are literally just a highway to communicate a signal message to another location. Dendrites, you can think about dendrites as being the receiving end of a cell body. So this is where the input of signals come from. So you think axons are output and dendrites are input. And then the second type of cell that we run into are called glial cells. And there's a ton of different types of glial cells. And once again, this is just the idea where if you're in the central nervous system, they're going to be called one thing. And then if you're in the peripheral nervous system, they're going to be called something else. And so essentially glial cells, they have multiple roles, but obviously with when you have axons that you don't want to be leaking the signal, They're going to be shielded in myelin. So they have a myelin sheath. And those glial cells are going to nourish, they're going to regulate the ions, the calcium, sodium, chloride ions that are in and are around that cell. They're going to offer support to the cell so it maintains its structural form. They're going to help with the transportation of waste and nutrients, and also with those neurotransmitters. So real quick, it's important just to distinguish and differentiate the difference between white matter and gray matter. And just to back up too, because I think I jumped ahead. So glial cells, just to make sure that we understand this, glial cells are not involved whatsoever in the sending of the impulse. The impulse is the impulse of the action potential. The impulse is primarily sent with the neuron itself. So the glial cells are not participating in the nerve signal sending, but they do insulate the wire. So it's almost like if you're flipping on your light switch, of course, the wire that runs from the switch to the light bulb is going to be conducting the signal, but the insulative material that goes around it isn't conducting the signal. It's just insulating it. Okay. When we talk about white matter versus gray matter, the best way to think about this is that white matter is white because it has the myelin sheath around it. The gray matter is basically the thinking part. So that would be the part where the dendrites and the somas are all kind of interconnected to each other, where they're processing information, but they're not sending it a radically long distance. They're just communicating back and forth between each other. And so gray matter is really just the thinking part, and the dendrites and cell bodies. And then the white matter is the sending part. So gray matter would be the collection of cell bodies and dendrites. But there's definitely not myelin sheaths there, which is why it's gray. And then also and of course, there's going to be glial cells there supporting that material as well. And with white matter, you're going to have all of these axons that have myelin sheaths that are kind of bundled together. It'd be like if you were to walk into the network room of a building, you would see a ton of like bundles of wires that are all kind of running through similar conduits in those buildings. And so that would be kind of like what the white matter is. It's the bundles of conduits that are carrying the nerve signals away from the cell bodies. And so, of course, the present of myelin makes them white. So just to kind of like give you a summary of that idea is gray matter is the thinking part. So that's cell bodies and dendrites. So that's signal processing. And then white matter is signal sending. So signal is sent through white matter because it's myelinated. Signal processing is done through gray matter. Perfect. And so this next slide is basically just reiterating the fact that you have two different naming systems based on which part or which location of the body you're in, but you're basically essentially talking about the same sort of things. And so when we talk about like groups of these cell bodies, so this would be like your gray matter that's in the central nervous system. In the central nervous system, we call those nuclei or nucleus if it's singular. So you have like a nucleus, which is like a bundle of in the central nervous system, you have a nucleus, which is a bundle of cells, like cell bodies that are all talking to each other. But if you move that same idea, that same structure into the peripheral nervous system, they call it a ganglion. And so you have a nucleus in the central nervous system, which is a bundle of cell bodies. And then in the peripheral nervous system, that bundle of cell bodies is called a ganglion. And they're essentially the same thing, but they have two different names depending on which location you're in. And the other thing to keep in mind too is, so that's talking about the cell bodies, like the gray matter. And then when you're talking about the axons themselves, like where you have those conduits that are running and transmitting signal, the white matter is going to be called, the bundles of those fibers in the central nervous system is called a tract. Whereas in the peripheral nervous system, it's called a nerve. And so we're really familiar, like, oh, you have this nerve that's running down your arm, you have this nerves in your spine and neck. But essentially, right, we're talking about tracts and nerves. So if it's in the central nervous system, it's a tract. If it's peripheral, like running down your limbs, that's going to be called a nerve. And so you can see here, right, these are the three different forms of nucleus. So we have a nucleus like an atom, we have a nucleus in a cell, and then we have a nucleus as a bundle of cell bodies, nerve cell bodies. And then this image over here is showing you how there's you have different tracks that are running in the central nervous system from one place to another. So you have this optic track, but then notice that once it switches to the eyeball, once it exits through the optic foramen, it becomes the optic nerve, because technically it's no longer part of the central nervous system. And so literally it's the same structure. It's just like once it passes through that hole, it becomes the optic nerve. But when it comes inside the brain, it's called the optic tract, even though they're exactly the same. So a taxi in America is a taxi. In London, it's called a lorry. So location dictates the term. All right. Let's see. So just to reiterate something, like you'll never see a ganglion in the central nervous system, but you will see a nucleus. So I went through and tested or looked at all the test questions. So I'm trying to reemphasize what you're going to see on the exam. All right. And so we have different functional divisions of the nervous system itself. So this is just central and peripheral together. And so you can think about these systems as one part of them is going to be processing, or one part of them is going to be transmitting information into the processing unit. And then one part of it's going to be the processing of that information. And then the other part of it's going to be the response of the processing. So it's like you get something in the mail, right? The mailman has to bring it to you. That's one part of the system. You read the letter, right? You process what you need to do with it. And then maybe you send a letter out back through the mail system. So that's kind of what we're going to run into with the nervous system. So we have sensation. So sensation is going to bring in the information from the environment. That can be internal and external. So that could be something like your stomach hurts or you got punched in the face. Both of them have a different origin, but they're both sensory input one's coming from a part of the body, from internal, the other one's coming from outside the body. So the next one is the integration. So you have incoming, that's the sensation. You have the integration, which is the processing unit of the nervous system, where it's taking in all the different sensations, it's computating them together, and then it's comparing them to past memories, to the emotional state, and then it's using the context of other sensory inputs to determine a proper response. And then the body's response comes after integration. So it's like, now what is the output? What do I have to do? And so you have voluntary responses, which you'll see like with skeletal muscle, and that's like the somatic nervous system. And then you're also going to have like involuntary responses, like smooth muscle contraction, your heart rate increases, some glandular tissue will secrete endorphins or other hormones that can be, I don't know, processed by other parts of the body. But the idea is so the response can be both conscious or unconscious. So like if you see a tiger, your heart rate's going to increase without conscious control. But when you're running, you're conscious of how your leg muscles are moving. And so that would be a combination in that response to the tiger, is that your heart rate has increased involuntarily, but your skeletal muscles are voluntarily contracting to get you out of there. All right. So now we're going to look. So if I was to take these three things, right, and I was to take sensation, integration, and response, and I was just to focus on the response portion and break that down, that's what we're going to do next. So we're looking just at the response portion. So I've received the letter, I processed it. Now, what are the divisions of response? So the first one is going to be the autonomic nervous system. So this is going to be it's the involuntary control part. So you've got the input coming in. Hey, maybe your body is hot, right? And so it goes through central processing and says, okay, in order to meet my homeostatic needs, I'm going to start sweating. And so you can think autonomic is homeostasis. So it's maintaining balance in the body. And so obviously like the outputs could be, smooth muscle, cardiac muscle, glandular tissue, that sort of thing. But it could also be stuff like sweat glands, and things like that. And then you have the somatic nervous system. So the autonomic was automatic. It's just for homeostasis. And then you have the somatic nervous system, which is for the most part voluntary control. So that's going to be things like skeletal movements. This could be reflexive. So like if you put your hand on a hot stove, you usually don't have to process that information in the upper parts of the central nervous system. It can happen in a in a cluster or a ganglion, if you will, that then immediately sends a response to move that skeletal muscle to get the hand off the stove. So that could be conscious or unconscious. It could be reflexive or it could be conscious. And then you have the enteric. And so something to remember with this one, with the enteric nervous system, is that's part of your gut. So when you're talking about like the and it's important to note that some classifications will put the enteric nervous system inside of the autonomic because you're not really ever consciously digesting your food. But there are parts of the enteric nervous system that seem to give it a mind of its own. So they kind of communicate with other parts of the nervous system. You have a great deal, for example, you have a great deal of, what is the name of that receptor in there? Oh gosh, now it's eluding me. I'll think of it later. It's the one that has to do with social hierarchy, and it has to do with like self-confidence and things like that, and that comes from your gut as well. And so the enteric nervous system doesn't actually need any signals from the central nervous system to do its job. It can digest food all on its own, and so that's why some people kind of place it in its own little system, and some people will place it inside of the autonomic nervous system. But the best way to think about the enteric nervous system is that it has a mind of its own. And a lot of people say, like, if you ever get a gut feeling about something, right, it's almost like you have two brains. Almost, it feels like you have one in your gut where you're like, I kind of am intuitive about this idea or this thing, or I'm getting a bad feeling about it, but it doesn't feel like it's coming from your head. It feels like it's coming from your gut. And so that's kind of how I like to split those things up. All right, so next we're going to look at the structure of a neuron. And so one thing to keep in mind is that you have a neuron, and when we talk about processes that come off of a neuron, we're only talking about dendrites and we're only talking about axons. So all of the processes of a neuron is the axon and the dendrites. Now, one thing also to keep in mind too is the axon, there's only one axon, but you can have multiple synapses from where the axon splits and integrates with different parts of the tissue. So you can have different terminal ends of the axon, but really there's just one axon that's carrying the signal from the cell body. An important part of this is the axon hillock. And so I labeled it on this diagram because it wasn't labeled in the book. But the axon hillock is really important because that is where the on or off switch comes from. So the axon hillock is like the nerve cell receives all this input through the dendrites and it processes all that information and it sends some chemical signals down here. And if they don't meet a certain threshold, this axon hillock works as a gatekeeper to the action potential. And so if it doesn't meet the requirement, the minimal requirement of stimulus, then it actually doesn't get activated. But if it does meet that threshold, then the action potential is triggered and it's sent down the nerve cell, down the axon, all the way to the synapses. And so the definition of a synapse is it's that specialized area of contact between neurons, but it's also, I mean, that is what it is, and that's what it will be tested on for this particular chapter. But, it's synapse is kind of like, it's between neurons, but you can also, sometimes that synaptic cleft comes into play where it's the gap between the synapse and the terminal end of the axon and its target tissue. So sometimes you'll see them used both ways, but for the most part, when we're talking about synapses, especially in like the central nervous system, it's talking about the contact area between neurons. And like I said before, there's only one axon, but there can be many corollaries that come off that axon and synapses. And then these nodes of Ranvier, these are interesting. What they do is they work through this process called saltatory conduction. So as the action potential comes down, you can imagine that every time these voltage gated ion channels are opening, they're leaking ions in and out of the cell, out of the axon, and then you got to pump them all back in. And so what these nodes of Ranvier are is they allow the signal to come through the myelin sheath without opening up any sort of gated channels. And then it repropagates the signal at these nodes of Ranvier. So it's almost like they jump. So the action potential comes down the axon, right? The last voltage gated channel here opens, sodium ions flood in here, and they just run down the axon without actually leaking. They hit here, which simulates, it's basically like a brand new action potential at each node of RANVA. And so it speeds the transmission up because it doesn't have to wait for every channel to open to get the signal. It almost jumps. It's like kapow, 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 all the way down. And so that's what it does. And that's done through a process called saltatory conduction, which we will address later on too as well. Okay, so now we're going to talk about several types of neurons. The most important one is going to be the multipolar neuron, and that's going to be the one that you're the most familiar with because that's one that has multiple dendrites that are attached to other synapses and other axons, and that's the one that can process multiple information. A lot of these other ones where we see the unipolar, that's really just taking a signal and then like a stimulus and sending it somewhere else. So we'll talk about these. So you got the unipolar or the pseudo unipolar. So it's got one process. So you'll notice here's the cell body and it only has one process. And then that process splits both into a dendrite and an axon. And so these unipolar neurons are very good at receiving sensory information and immediately transmitting it down the axon to its target synapse. Usually it's running it up towards the central nervous system for processing. And so the unipolar neuron, if you'll notice, right, the actual cell body itself is not involved in whether or not that signal gets sent. It's just there for support, right? This one's here for support. It's not controlling whether the signal was strong enough to get sent. So the stimulus comes in and pow, it's gone down to the axon, down to those target tissues. And with the bipolar neuron, you've got two processes. So you still have a dendrite and an axon. These aren't very common, right? These are, like it says, found in the eyeball and the olfactory system. But this one does put the cell body between the two. So the dendrite comes in, you get some processing, and then an output. Whereas when you have the multipolar, you've got the cell body that bring all the dendrites come into the cell body for complex processing, and then a decision is made whether or not that signal gets sent down the axon to its target neurons. And so the multipolar one is the most common because it allows multiple inputs from different sources in central processing. So it allows the complexity of the system itself to just exponentially increase the more of these that you have. All right. And so, yeah, the unipolar neurons are always found in ganglia. There you go. And then there's your dendrites. Perfect. Okay. All right. So moving on to that was talking about neurons. So remember that we split the two different cell types that we find in the nervous system are neurons and glial cells. And so next we're going to look at the different types of glial cells. Now, there are 6 types, right? But 2 of them are pretty much the same. They have slightly different structures. And so in the central nervous system, you have astrocytes, and their equivalent cells in the peripheral nervous system are satellite cells. And we'll talk more about their basic function, but we'll go into detail a little bit more about their overall function. And then same thing for the oligodendrocyte and the Schwann cells. These 2 ones are very, very similar, but they have slightly different mechanisms for action. And then only in the central nervous system do you find the microglia and the ependymal cells, because that's where you have the cerebral spinal fluid. All right, so we're going to dive into these in a little bit more depth. So in the central nervous system, we'll talk about the central nervous system cells. That's these ones right here, the astrocytes, the oligodendrocytes, the microglias, and the ependymal cells. We're going to talk about those more in depth now. So the astrocyte, when we look at it under a microscope, it kind of has a star shape, and we've seen these before, like on tissue slides and things like that. And it has multiple roles. So and I'll break these roles down into just simple words. It's going to be support, protect, repair, and nourish. And it's the functional equivalent to a satellite cell. So astrocytes protect, support, protect, repair, and nourish. So here they are. They create structural supports. They surround the cell and kind of keep it in its area so it's not bouncing around and getting damaged. And then it's it's supporting, protecting. So it's going to maintain like like the calcium ions, the sodium ions, the potassium ions. It's going to maintain the chloride ions. Also the amount of water that's coming in and out. So it's kind of filtering what's being available to the cell based on the cell's needs. And so you could think about these as like the astrocytes are like the butlers to the neuron. The neuron doesn't do anything except send signals and process signals. And so it's got such a busy job just doing its own job that it needs a butler to take care of all of the other needs around the house. So it needs the butler to fix, you know, the windows when they break. It needs the butler to, you know, build a new bedroom or something. I don't know if that's something a butler does, but and then it, you know, if it needs water, you know, it's like, hey butler, go get me my water. Okay, here's your water. Hey butler, I need some more neurotransmitters, or I need you to break down these neurotransmitters. So the astrocytes are really just like the butlers to the nerve cells. And then they're also going to help regulate how much sugar the cell is getting. So again, it's this nurse cell. It's taking care of the neuron. And so it's going to filter things out. It's going to take the waste products, put them in the bloodstream. It's going to filter the blood and give just the right amount of stuff to the neuron. And then, of course, it'll help to damage the neuron. But once the neuron dies, the neuron's dead forever, and you won't ever get that back. And again, the blood-brain barrier is just this idea that you're filtering the stuff that's getting to the neuron because they're so important to supporting life. And so the next glial cell that we have is the oligodendrocyte. And so this is the one that myelinates. So you can think astrocyte is the butler. The oligodendrocyte is the myelin. So that's the insulation around the wire. And so here's the difference between the oligodendrocyte and the Schwann cell. The oligodendrocyte is going to be able to insulate more than one axon with the myelin sheath. So if the oligodendrocyte is adjacent to several axons, it can actually provide the myelin sheath to those multiple axons, whereas the Schwann cell won't be able to do that. And then you also have the microglia, and you know, if you remember our lecture from Steven, when he came in, I was talking to him before class, and he was saying, hey, he's like, every single tissue is going to have their own form of a macrophage, like an immune cell that actually eats up things that are not supposed to be there. And so these microglia are basically like the macrophages of the nervous tissue. They're basically derived from the immune lineage. And so they're able to kind of go around, and they can go in and out of through the blood-brain barrier, eat up pathogens, and just make sure everything's staying nice and clean, and you're not getting any sort of infections in your nerve tissue. And then you have the ependymal cells, and these ones just kind of, they're specialized cells, like endothelial cells that are lining the blood-brain barrier, and they're producing the cerebral spinal fluid and filtering it. So they're filtering the blood to make the cerebral spinal fluid. And that's all you really need to know about those for now. And we'll talk about them more probably when we get to module 4. But those are the 4 main glial cells of the central nervous system. And then in the peripheral nervous system, I don't think I had a picture here. So in the peripheral nervous system, we actually have an image here of these different cells and what they're going to be doing. So remember that in the peripheral nervous system, satellite cells are going to be the butlers to the neurons that are in the peripheral nervous system, whereas the astrocytes were the functional equivalent in the central nervous system. So astrocytes, satellite cells, butlers, notice how they're surrounding the cell, and they're just like, here, let me take care of that for you. Let me get you some water. Let me break down your neurotransmitters. Let me do anything I can to help you. And so again, you have the satellite cell surrounding it, protecting it, offering supports, and many of the same functions as the astrocytes. The Schwann cells, right? These are going to be the ones that are myelinating the sheath, the axon, or they're creating the myelin sheath around the axon. And the only difference really here is just that in the peripheral nervous system, the Schwann cells can only cover one axon, whereas the oligodendrocytes can actually cover multiple axons, which, if you really think about it, in the central nervous system, you have a lot of axons that are running from one location all the way down a spinal column. And so it makes sense to kind of like say, okay, energetically, what makes the most sense here, to have one cell that's supporting multiple tissue, like multiple structures, or individual cells supporting individual structures. And if you're trying to be evolutionarily conscious of your energy expenditures, you're going to just go, okay, if I can have one cell do the job of multiple cells, and it's just as functional, that's probably going to win out in the arms race of energy expenditure. And so that's the Schwann cell, and the peripheral nervous system is going to be covering the axon. All right. And so I've got this a couple more slides, and then that should be about it. Perfect. Okay. So we're going to just talk through how information, in a very general way, we're just going to talk about information and how it's transmitted through the central nervous system, or through the peripheral nervous system to the central nervous system and back. Now, I don't need you to memorize anything that's on this slide, but I do want to talk through just the image itself and how things are being processed, the inputs and the outputs here, okay? And so you have these sensory receptors that are in the skin, right? We know these to be like the Meissner's corpuscle and the Bassinian corpuscle, things like that are in the skin that can detect thermoreceptors, things like that. And so they're going to receive a signal, and that signal is going to be propagated all the way here. And this is basically the cell body, right, of this sensor right here. This is the cell body all the way back here. And then it's going to transmit down its axon all the way to the central nervous system. So this right here, this sensory neuron, is this sensory neuron. So it's picking up the signal here and transmitting it all the way here to the cell body. And then it's passing that signal on, right? And in these ganglias, what you're going to see is like there's some limited processing going on here. So if it's like I touched a hot stove, it might get to here, the spinal cord, and then bam, immediately send a motor output, like move that hand before it catches on fire. But if not, if you're just testing like the temperature of water, it'll send the signal up here where the sensory axon is going to hit this interneuron, and the interneuron takes that signal and sends it to the thalamus. Now this is on the test, and so I want to make sure that this is emphasized here. The thalamus acts like the relay station for the signal. So the signal comes in, right? It gets all the way to the thalamus, and the thalamus is taking in multiple sensory inputs all at the same time. And so then it's going to relay that information up here to the cerebral cortex, where the processing happens. Cerebral cortex is then going to make a decision on the output. And then from the motor cortex, it's going to run all the way down to the spinal cord, and then all the way back to the tissue and the skeletal muscles that are there. So if the water is super hot, or maybe it's just warm enough, and it's time to move the hand, so you receive the warm signal. Okay, yeah, it's time to get in the shower. Okay, thalamus sends that signal. Now you're processing. Okay, the water is warm enough. I'm kind of awake. It's time to get in the shower. Okay, I'm going to move my hand to open the shower door. Okay, so I then move my hand, and then I open the shower door, or I move my hand to turn up the hot water a little more, the cold water down a little more. And so the thalamus acts as sort of this sensory hub that's processing and sending the information to the cortex so that it can do its job of really computating what's the best move forward from here. And so, like I said, it filters and sends the relevant information to the cerebral cortex, and then the cerebral cortex is the processing station. So that is, when we think of the brain, we're thinking really of the cerebral cortex. And so that's computating all these complex signals that are coming in from the thalamus with situational data points, memory, emotion, that sort of stuff. And then it's making a decision on what to do. And don't worry, like, I know this is a big fat like paragraph here, but the precentral gyrus of the frontal cortex is the motor cortex. And so that's going to send the signal down the upper motor neuron to the lower motor neuron, which is going to activate muscle fibers on the target muscle that's going to move that limb. And so that's really just, I wanted to give you guys just like a really brief overview of what's happening, right? You have the sensory, comes to the ganglia, right? The nerve body sends a signal down the axon to the interneuron. Interneuron sends that signal to the thalamus. Thalamus is taking in multiple points of data from sensories, sends it up to the cerebral cortex. Cerebral cortex is also taking into context like time of day, location, memory, emotions, processes it, and then it sends the signal to the motor complex. That's the precentral gyrus of the frontal cortex. So the motor cortex is then going to send the signal down the motor neurons to the skeletal muscle to have them move. And so that's the important parts to know there. So we're going to talk a little bit more about this precentral gyrus of the frontal cortex because it's super fascinating. At least I think it is. And so another word for this is the homunculus. The precentral gyrus of the cerebral cortex is it's the motor homunculus. And so what's really cool about this motor homunculus is this little image here shows you each size of the body here is proportional to the number of motor units that are in the nervous system for that part of the body. So it's like your mouth, your lips, your tongue, your fingers, right? Those are all going to have a lot of motor neurons going to them because you can change your facial expressions right around your mouth. Like there's so many different like complex ways you can do that. And then your fingers, you have so much fine little motors that are going on here that they're going to take up a larger proportion of real estate in the actual brain itself. And so that's one of the most fascinating things about the primary motor cortex is that the amount of space that's taken up here is directly proportional to the precision and complexity of the motor units. So it's like, so for your eyes, nose, and face, because have so many different motor units, they're going to take up a large part of the real estate. And then same thing for the hand. Like the hand is such a small proportional part of the body, but takes up such a large proportional part of the cortical space there in that motor homunculus or in the primary motor cortex. God, it's hard to talk. And so, like we said here, hands, fingers, lips, and tongue, things that are very sensible or have a lot of sensory, but also a lot of motor units are going to have the highest proportional real estate. And so the way I put this was like, think about playing an instrument like a piano or a guitar or something like that, is you have to have such finely tuned abilities to move those fingers in such a complex way and very refined way that it's going to take up a lot of real estate inside the brain itself to do that. And so that's kind of where we're going to end up here. Is that's it for today. So that's 40 minutes of lecture. I know I didn't break it up for any questions or anything like that, but that's where we're going to stop, and we'll pick up with the action potential on Thursday. So I hope you guys enjoyed this whatever online lecture in place of our Halloween one. And I will see you guys on Thursday. So adios.